Section 1. Introduction: The music of life. Life is a symphony of molecular processes. Protein synthesis stands out as the grand orchestra of the cell, translating the genetic blueprint into functional molecules. These proteins build our cells, catalyze reactions, transport molecules, and defend us against invaders. Understanding protein synthesis is about appreciating the flow of information from genes to proteins. This process unfolds in two major movements: transcription and translation. Section 2. The score: transcription. Imagine a vast library filled with countless volumes. This is the cell nucleus, and the volumes are our chromosomes, made of DNA. Each gene holds the instructions for building a specific protein. Transcription begins with RNA polymerase, a molecular machine that unwinds the DNA double helix. RNA polymerase reads the sequence of nucleotide bases: adenine, A, thymine, T, guanine, G, and cytosine, C. As RNA polymerase reads the DNA sequence, it adds complementary RNA nucleotides, building a single-stranded RNA molecule. This newly synthesized RNA molecule is the messenger RNA, mRNA, carrying the genetic message from the nucleus to the cytoplasm. Before the mRNA can leave the nucleus, it undergoes crucial processing. The cell adds a protective cap to the beginning of the mRNA and a tail of adenine nucleotides to the end. Once processed, the mRNA is ready for its journey out of the nucleus. Section 3. The orchestra translation. The mRNA exits the nucleus and enters the cytoplasm, encountering ribosomes, the orchestra of protein synthesis. Ribosomes bind to the mRNA and begin translation, converting nucleotides into amino acids. The mRNA carries the genetic code in the form of codons, groups of 3 nucleotides specifying an amino acid. Transfer RNA, tRNA, acts as the interpreter between mRNA and proteins. Each tRNA molecule carries a specific amino acid and has a 3-nucleotide anticodon complementary to an mRNA codon. As the ribosome moves along the mRNA, it matches each codon with its corresponding tRNA anticodon. The tRNA delivers its amino acid to the ribosome, adding it to the growing polypeptide chain. This process continues until the ribosome reaches a stop codon, signaling the end of the protein. The completed polypeptide chain is released, ready to carry out its role in the cell. Section 4. Regulation. Fine-tuning the symphony. The cell tightly regulates protein synthesis, ensuring the right proteins are produced at the right time and in the right amounts. One level of regulation occurs at transcription, where transcription factors can enhance or repress RNA polymerase binding. This control mechanism influences the amount of mRNA produced for a particular gene. Another level of regulation takes place during translation, where various factors control the initiation of protein synthesis. The cell can also regulate protein synthesis by controlling the lifespan of mRNA molecules. Some mRNAs are rapidly degraded, while others persist longer, allowing for continued protein synthesis. External factors like nutrient availability, hormones, and stress signals can also influence protein synthesis. These cues trigger signaling pathways that modulate the activity of transcription and translation factors. This fine-tuning ensures the protein synthesis machinery meets the cell's changing needs. Section 5. A NEAT example: cracking the code. Let's apply our knowledge to a typical NEAT exam question. A segment of a DNA molecule has the sequence 3' TAC GCG ATC 5'. What would be the sequence of the corresponding mRNA molecule? Option A: 5' AUG CGC UH 3'. Option B: 3' AUG CGCC UEG 5', Option C, 5' ATG CGC tag 3', Option D, 3' ATG CGC tag 5'. RNA molecules are synthesized in the 5' to 3' direction, reading the DNA template strand from 3' to 5'. Flip the given DNA sequence to represent the template strand, 5' CTA GCG GTA 3'. Replace each DNA base with its complementary RNA base, remembering that uracil, U, replaces thymine, T, in RNA. From DNA, 5' CTA GCG GTA 3' to mRNA, 3' GUCGC COL 5'. Flip the mRNA sequence to represent the conventional 5' to 3' orientation. The correct answer is A, 5' AUGCGC UEG 3'. Section 6. Mutations. When the music changes. Mutations, changes in the DNA sequence, can disrupt protein synthesis, leading to abnormal proteins or altered protein levels. Mutations can arise from errors during DNA replication, exposure to mutagens, or spontaneous chemical changes in DNA. The impact of a mutation depends on its location and nature. Some mutations may be silent, having no effect on the amino acid sequence. Missense mutations result in the substitution of one amino acid for another, potentially disrupting protein function. Nonsense mutations introduce a premature stop codon, truncating the protein. Frame shift mutations alter the reading frame, leading to a different amino acid sequence downstream. Mutations can lead to genetic disorders or increase the risk of diseases like cancer. However, mutations also drive evolution, providing genetic variation and adaptation. Section 7. Beyond the basics, exploring the depths. For aspiring NEET candidates eager to delve deeper, here are some avenues for further exploration: post-translational modifications, protein folding and misfolding, protein targeting and degradation, regulation of gene expression, and emerging technologies. Post-translational modifications influence protein function, localization, and interactions. Protein folding is crucial for function, and misfolding can lead to disease. Chaperone proteins assist in proper folding and prevent aggregation. Signal sequences direct protein trafficking to the correct destination. Proteins are selectively degraded when no longer needed or damaged. Understanding gene expression regulation is crucial for comprehending development, differentiation, and disease. Advances in genomics, proteomics, and bioinformatics provide insights into protein synthesis and its regulation. Techniques like CRISPR-Cas9 and ribosome profiling revolutionize our understanding of this process. Section 8. Conclusion: The Unfolding Symphony. Protein synthesis is a fundamental process that underpins all life. The cell orchestrates a complex symphony of molecular events, culminating in the production of proteins. This intricate dance of transcription and translation ensures the right proteins are produced at the right time and in the right amounts. Mastering protein synthesis principles is about appreciating the elegance and complexity of life at the molecular level. Understanding how information flows from genes to proteins and how mutations can disrupt this process is crucial. Protein synthesis is a dynamic symphony, constantly adapting to the cell's needs. Embrace the challenge of delving deeper, asking questions and seeking answers. The journey of understanding life's molecular symphony is just beginning.